In this video, we will discuss about one of the immune response, that is antiviral interferon response, and this is one of the innate immune response, and compared to the other innate responses, it is also related to the adaptive immune response. And in the case of SARS-CoV-2, it has been found that the levels of this interferon goes down drastically compared to the normal levels. So one of the ways by which this SARS-CoV-2 virus is more infectious is that it lowers down the interferons levels uh, through some mechanism. So in this video we will discuss how this interferon actually normally functions, and how it will get activated, or how it will get triggers uh, within the within the intracellular, or how it will get triggers in the intracellular parts of the cells. So there are two. It has been the interferon response has been divided into two different sections. First one is interferon induction, and second one is interferon action. So the process initially begins with the interferon induction. In this case, when the several immune components, when it detects any kind of harmful things within the host, through such as this PAMP, that is pathogen associated molecular patterns, or some other immune mechanism similar to PAMPs. Whenever it detects any foreign harmful and particles, it will generate the or it will triggers the interferon inductions through three different pathways. The PAMPs, or immune other part of immune system similar to the PAMPs, will trigger interferon inductions through three different pathways. First one is toll-like receptors and RLL pathways and C-gas pathways. So in the case of these toll-like receptors, what will happen is that the viruses along with the viruses will get will enter into the cells, and when the virus interacts with the toll-like receptors, and thereafter it can go to different signaling pathways that has been shown here. If it goes through this uh, MyD 88 pathways, it will then goes and finally triggers NF kappa beta pathway, and this NF kappa beta pathway with upregulates the pro-inflammatory cytokines, and these pro-inflammatory cytokines will leads to this pre-mRNA formation, and after that this signaling molecule, or interference, this pre-mRNA of the interferons formation, and after that this normal process, that is maturation of the mRNA, and then due to the signaling SRP, that is signal recognition particles, it will then directed in the extracellular matrix, and only a slight changes occurs if the uh, viruses enter into the cells through these RLR pathways. In this case of RLR pathways, the RNAs of the virus directly entered into the cells, whereas in the case of toll-like receptors, whole virus entered into the cells, and thereafter lysis of the outer membrane occurs. Whereas in the case of RLR pathway, only the RNAs and the PAMPs that detects those foreign RNAs enters into the cells, and thereafter through this RIG1 pathways, it will then move to MAV pathways, and then it will trigger this IRF3, and this IRF3 IRF3 then go inside the nucleus, and the phosphorylation of the IRF3 will triggers pro-inflammatory response, and in when the IFN induction is occurs through this C-gas pathway, what will happens in the case of C-gas pathway, when the when the foreign harmful particles has the DNA component, then it will go through this C-gas pathway. In the case of this C-gas pathway, it will trigger this C-gas and then the sting pathways, and thereafter it will join, or it will the pathways will get merged to this RL pathway, that is TBK1 and IKE complex formation occurs, and it will trigger IRF3, that will lead to the pro-inflammatory cytokines, and that will all this will finally trigger the formation of interferon 1s, and all this red component that is mentioned is to prevent the interferon formations. So viruses could target uh, at any of one of these sites to prevent the interferon productions, or we can say interferon inductions. And after that, when the interferons are produced, it will then go into the um, appropriate receptors such as type 1 interferon receptors or type 3 interferon receptors, and that will triggers further downstream signaling pathways that leads to the complex formation of this IRF9 and STAT1 STAT2 phosphorylated, and this complex will then go into the nucleus, and then it will trigger the pre-mRNA formation, and after that mRNA formation, and because of the signal recognition particles and these cytokines that is responsible to take care of the harmful particles will then move into the cytoplasm or into the extracellular matrix based on the SRP signal, that is signal recognition particles, and then thereafter it will take its action for which it is finally been designed or produced. So this is how and in this case also there are several steps where uh, these intercellular signaling pathways can be disrupted. That has been shown here the using the red markers. So this is so in general terms, this is how the IFN induction and action take place, and this all the red mark point could be one of the possible approaches can be used by any viruses to avoid this interferon uh, induction and interferon production or actions. So I hope you got an idea how this antiviral interferon response works. And if you need to understand more about this antiviral interferon response and uh, what happens when the person gets infected with the SARS-CoV-2 to this interferon response, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.